Hi, today we'll be talking about the endomembrane system. The endomembrane system consists of a couple organelles. For example, it consists of the nucleus, the rough ER, the Golgi apparatus, to some extent the lysosomes, and the plasma membrane. The endomembrane system is a key system in protein synthesis. As seen here, it is able to synthesize secretory proteins, hydrolytic enzymes, and extracellular proteins, as well as detecting failed proteins which are then destroyed by the lysosomes. We'll first be discussing the secretory protein. It begins in the nucleus, where we have a gene, and this gene is transcribed into an mRNA, or a messenger RNA. This messenger RNA travels through the rough ER lumen and arrives at a rough ER-bound ribosome. This ribosome then turns it into codons, which then turns it into a secretory protein, for in this instance is insulin. This insulin is packaged into a vesicle and sent out to the Golgi apparatus. Here, the vesicle arrives at the cis face of the Golgi apparatus. The cis face is the receiving face of the Golgi apparatus. This vesicle coalesces with the Golgi apparatus, releasing insulin within the Golgi apparatus. Now, the Golgi apparatus does a couple things to this insulin. For starters, it's able to modify it slightly to better its abilities in doing its job, as well as being able to tag the insulin, which acts a lot like zip codes within the body, i.e. it being docking sites, just as how mail is to us delivered with the location. This insulin is then packaged into a vesicle, which then goes to the plasma membrane, and through exocytosis, exits the vesicle and into the extracellular matrix, and is then sent out to do its job. Next, we'll be talking about hydrolytic enzymes. Hydrolytic enzymes are key things, they're key proteins to destroy other proteins, mainly misfolded proteins. Hydrolytic enzymes are also the important factor in creating lysosomes. Much of the similar process as to insulin, it starts with the DNA being transcribed into mRNA, traveling through the rough endoplasmic reticulum lumen, which is then in this case turned to a hydrolytic enzyme or hydrolytic protein, packaged into a vesicle, and sent off to the Golgi apparatus. And much of the similar process arrives at the cis face of the Golgi apparatus, coalesces, slightly modified by the Golgi apparatus, then tagged, and on the trans face is packaged and becomes a lysosome. Now, in some instances, lysosomes are created by the Golgi apparatus. There are some instances where the hydrolytic enzyme combines with another lysosome to increase production or decrease the amount of lysosomes present within the body, but most times lysosomes are created through the Golgi apparatus. This lysosome is then left into the body to do its job. Next, we'll be talking about the misfolded protein. Misfolded proteins are extremely bad to the body, as it can create issues, for example, a prion disease, which is fatal to the body, as misfolded proteins will cause other proteins to misfold. So it is extremely vital for the cell to kill or at least rid of misfolded proteins. Much of a similar process, the gene is then transcribed into mRNA, but in this instance, the mRNA seems to have some sort of issue with it, some transcription error or something of the sort. Traveling through the rough endoplasmic reticulum, it is then unfortunately made into a misfolded protein. The rough ER immediately detects that this protein is misfolded and then calls over a lysosome, which then uses its hydrolytic enzymes, which is created by a similar process and packaged by the Golgi apparatus, and kills or destroys the protein and recycles it for later use. Next, we'll be talking about extracellular glycoproteins, or in this case, antigens and other receptor proteins. These are important for the cell as it's able to communicate with other cells as well as create structure within the extracellular matrix and able to connect with cells to share nutrients and of the sort. Much of the similar process as to every other protein that has been created through the endomembrane system goes to a membrane-bound ribosome, transcribed into this case extracellular protein packaged into a vesicle and sent off to the cis phase, the Golgi apparatus. The Golgi receives this protein, coalesces, slightly modified, is then tagged, packaged on the transphase, goes to the plasma membrane, and through exocytosis is able to be released into the body. However, in most instances, through exocytosis, the moment it is released, the protein binds to the plasma membrane. In this instance, for better visuals, as we can see, it's binded to the plasma membrane. Thank you for listening to my presentation, and hopefully we'll learn something together.